Hi, so I'm Megan, and I'm going to present my GIS project where I wanted to reroute shipping vessels to avoid zooplankton. So we all know what zooplankton are. They refer collectively to the species that make up the small animal component of the planktonic community. So they can either be adults or larval staged organisms. Basically, they're just really important for transferring energy between the photosynthesizing phytoplankton to the larger secondary consumers. But why should we care about zooplankton? Plankton specialists around the world have really been stressing plankton declines within the 21st century. And some articles have even quoted a 50% decline in overall plankton biomass within the last 2 decades. And these are global trends all over the world. So the environmental mechanisms of zooplankton declines are pretty well understood within academia. However, recent work has shown that maybe climate variables aren't the whole story. And that's where shipping comes in. So 90% of our global resources are transported by ship, and we have over 55,000 actively trading merchant ships worldwide. Unlike cars, ships follow network routes to get from port to port, and that's what you see on the globe on the right here. This is just 1 year worth of shipping routes, and it's crazy. You can see outlines of the continents without even having to draw those continents. And along these routes, the high ship densities threaten marine wildlife. And among the various ship-caused threats, noise pollution and collisions have been consistently proven to impact large mammal survival, and recent work has shown that zooplankton are similarly affected. How will this project help? My main objective was to provide some preliminary work towards the design of optimal shipping routes that avoid important marine wildlife. And so zooplankton was just my species of choice. So I thought there was 3 main goals that I needed to achieve in order to provide valuable knowledge on this topic. The first goal was to make use of long-term data to identify areas of abundant zooplankton. Then I wanted to determine the spatial relationships between zooplankton abundance and shipping density. And then my third goal was to create these optimal shipping routes between major ports that avoided the identified with zooplankton hotspots. So I couldn't do this for the whole ocean. So I chose to look at the ocean off the east coast of the United States and Canada, from about Virginia to Nova Scotia, just like you see on the right here. I primarily chose this region because of zooplankton data availability. And then also what you can't see in this map is I created a 5-kilometer square grid throughout the entire study area, and I used that as the resolution of all of my analyses. So if you look at the study area, it looks like a buffer, which is exactly what it is. It is a 250-kilometer buffer offshore. I chose this value because there's something called the Northeast US continental shelf that runs through the entire length of the study area, and it goes up to 200 kilometers offshore. So I wanted to be able to capture that in my study area, mostly because continental shelves are really well known for having high zooplankton abundance. So then it brings me to my first goal, which was finding those areas of abundant zooplankton. So my zooplankton abundance data came from the Ecosystem Monitoring Program surveys that are run by the NOAA between 1977 and 2017. That's not what you see on the right, but I'll explain that in a second. So the entire data set was used to find long-term consistent regions of high zooplankton abundance. And I used chlorophyll A concentration as a predictor variable because the original data came as points, but I wanted to create a continuous surface of abundance over my study area. Chlorophyll A was sourced from NASA, and it's what you see on the right. It is a concentration, an annual average from 2009 to 2013. And it's used as a proxy for phytoplankton abundance, which is the major food source of zooplankton. And previous work has shown that predictions of zooplankton abundance can be accurate using chlorophyll A data and also using regression methods. So it was just a really easy to get set of data that was handy to use for this project. Some important things to remember are that chlorophyll A concentration is highest around the shorelines. So to predict the zooplankton abundance over the gaps, I used Ebk regression, which is a method of interpolation. There's lots of methods of interpolation I could have used, but this was like a straightforward one that had an explanatory variable I could input. And again, that explanatory variable was chlorophyll A concentration. So the map on the right shows those final zooplankton abundance predictions, where these dark green areas and the light green areas are the higher zooplankton abundance and the gray are the lower. There's two main characteristics of this map that I want you guys to remember. The first is that, much like the chlorophyll layer, abundance predictions are larger in the northern region of the study area up here. And the second thing I want you to remember is that, much unlike the chlorophyll layer, the abundance predictions near the shorelines were very low compared to other areas of the study area. That caused some problems later, which I'll mention. So that brings me to the end of my first goal. I identified the areas of high zooplankton abundance. So then I wanted to assess the spatial relationships between those areas of zooplankton abundance and shipping density. The purpose of exploring these relationships was to determine whether there was a statistically significant reason for avoiding zooplankton within the study area. So should I even be creating optimal shipping routes for my study area? So my shipping density data was from 2009 to 2013, and it was sourced from Halpern et al. Their version of the data was a simple linear average of a bunch of layers that were created by a guy named Walbridge, who created a shipping density algorithm. And so the map on the right shows the average shipping density values, but I summarized them I summarized the average value within each of those 5 kilometer grid cells that I created. Just take note of the high shipping density areas of the study area. So the southern region seems to have a lot more shipping than the northern region. So once that shipping data was finally contained, which took me about 2 weeks, I grabbed that zooplankton abundance data that I interpolated from previously, and I summarized it within the same 5 kilometer square grids. And then I ended up actually further classifying the zooplankton data into 10 categories, from like lowest to highest zooplankton abundance, and that just made it so that my computer could handle the analyses a bit better. So to assess the relationships, I threw the zooplankton and the shipping layers into a tool called the local bivariate relationships. Basically what this tool does is it determines the shape of the abundance density relationship in each of those 5 kilometer square grid cells by running a regression with each cell's 50 closest neighbors. And then it uses like R-square and p-values to classify these relationships into one of these 6 categories. So the red and kind of salmony color are linear and nonlinear negative relationships, meaning that more shipping density would have lower zooplankton abundance. And then the dark and light green are positive linear and nonlinear relationships. My results for this were pretty surprising. I was expecting a lot more negative relationships. But as you can see, there's not as many. There's some more in the north, and they're also scattered through the study area with lots of non-significant regions throughout the study area. But I think there was 3 main reasons why maybe I didn't get the results I was expecting. The first is that the zooplankton data was collected on ships, and those would have shipping impacts that would have affected the abundance observations. The second possibility is that the resolution of my analyses masked more localized effects, which are maybe more important in this context. And then maybe there was also an external variable that was influencing zooplankton abundance that I was just not capturing in the shipping density. So because there was a large uncertainty in my results for this, I decided that any significant negative linear or concave results were deemed adequate reasoning for me to go ahead and continue creating optimal shipping routes. So that's what I did. And I created these optimal shipping routes that avoided zooplankton by using the least cost path tool. So this tool essentially finds the shortest path between 2 points with the smallest accumulated cost. So to create this least cost path, I have to assign the cost of traveling across individual cells of the study area. So I need to say that certain regions of the study area, I want those paths to avoid. They're not optimal to move through. And other areas of the study area, I need to say, I want to encourage the paths to move through those areas. And so we're going to call the result of this cost assignment the cost surface. Layers incorporated in the cost surface need to be reclassed to a common scale so that cells with larger scaled values are quote unquote more costly to travel through. So the cost surface that I created for the project incorporated 3 reclass layers. The first layer is that Zooplankton Abundance, which I interpolated, and I also previously reclassed into 10 categories, where cells with a category value of 10 have the highest zooplankton abundance. The second layer I incorporated was the shipping density layer that I had summarized as well. And then I further reclassed the shipping density into 10 categories, much like the zooplankton abundance layer. However, the cells with the higher category values were actually the lowest shipping density values. And then the third layer I incorporated was a newly created buffer layer, which is what you see on the left. There's a 25-kilometer offshore buffer region, which is this red section. And I assigned that a category value of 10 so that it was very expensive to travel through these red areas. And then the remaining regions of the study area up to 250 kilometers offshore, which is this gray area, were assigned a category value of 1. So it was really cheap to travel through the rest of this study area. Reclassing the layers in this way incentivized traveling through cells that were regions with low zooplankton abundance, that were commonly shipped in the past, and that were offshore. So these are the 3 characteristics that I wanted my shipping routes to move through. And then each reclass layer also had to be converted to a 5-kilometer square resolution raster. And those were what were used to create the final cost surface. So on the left, you'll see the general purpose cost surface that was used for every single least cost path to follow, where the higher the cost, which is these darker red areas, the less incentive there was to travel through those cells. So this cost surface, like I mentioned before, was created by combining the zooplankton abundance, the shipping density, and the buffer rasters. And I combined them using weighted sums, where 70% of the combination represented the zooplankton abundance layer, and the remaining 30% represented the shipping and buffer layers equally. So I had to have zooplankton abundance contributing the most to this cost surface because it is a zooplankton project. There's 2 primary characteristics of this map that are going to be important to remember. The first is that it is less optimal to move through this northern region. There's a lot more dark red, and that the northern shores have really high cost values. So we really want to avoid the northern shores, it seems. The second characteristic to remember is that the southern region is more optimal to move through, and that the southern shorelines have some areas with really low cost values. So it seems that the southern and the northern regions are quite opposite in this sense. So the cost surface that you saw on the previous map had to be used to create a cost distance surface for each individual pair of shipping ports. By creating a cost distance surface, I'm essentially just accounting for the distance in the cost of traveling between ports, because I don't just want the least cost path. I also want a short path. I don't want to like go all the way around to avoid every single area of high zooplankton abundance. I want it to be efficient as well. So once that cost distance surface is made, it's simply just inputted into the least cost path tool, which outputs a polyline route between specified start and stop points. So I had to do this for each individual set of points for each route that you see on the left. So yeah, the left shows those final least cost polylines that I created. And I'm not going to talk too much in detail just for like sense of time today. But just really take note of the distance that the paths are from shore. They seem to be pretty close most of the time to shore. Some other important characteristics of these least cost paths are that they were only created between large shipping ports, and that's a classification system from the shipping industry. Also, some ports were outside of the study area, so I used a nearby location as the start-stop point. So for example, if we go to Halifax, here is the actual port of Halifax, and this is the point I used for the start or stop of routes coming or going to the port of Halifax. And then the last thing is that some ports were really close to one another, so I only created one path for those neighboring ports. The most extreme example of this will be, this is the port of Norfolk, this is the port of Baltimore, and this is the point I used as the start or stop point for both of these ports. This represented both of these ports. So the results from this project show a glimpse of the potential for designing routes that are both feasible for the shipping industry and more beneficial for the marine life of the traveled waters. If you're interested, I split up each route by its destination, and you can look at some different characteristics of them. But again, just for sake of time, I'm not going to go too much in detail. However, there are 3 take-home messages or consistent characteristics of all of the routes that I created. The first is that routes to or from more northern ports traveled through proportionally more highly abundant zooplankton regions than the mid to southern ports. So it seemed that if you were moving to northern ports like Halifax, you would end up having to go through a lot more highly abundant zooplankton regions than if you were moving to more southern routes like Norfolk or Baltimore. The second take-home message is that as distance between the ports decreased, routes traveling on the shoreline became more common. So it seemed that offshore travel only occurred if your ports were really, really far apart in the study area. And then thirdly, there were some consistent resolution errors in the creation of my routes, which caused some travel across islands or peninsulas. And obviously this is not realistic or feasible, so that definitely could have been changed in the future. So obviously there were some major limitations that need to be revisited in future work. The first limitation was the accessibility of the shipping data. So most of the shipping information was exclusive to the shipping industries or shipping companies who collected the data, which was kind of frustrating in finding good information. Had this been different, I could have specified more parameters in the cost distance tool to make it more accurate, or I could have compared my optimal shipping routes to actual pre-existing routes. But the extent that I could do this type of comparison is kind of shown here. I could just look at the density graph and say, oh, this doesn't really follow closely to what regions of high shipping density are, but this isn't a very accurate comparison. Also relating to the shipping data was my own lack of common knowledge about the shipping industry. I tried to incorporate the feasibility of my routes, but I was just unaware of the major motivations for the choice of major shipping routes. The second big limitation was the bias in the zooplankton interpolation. So because the zooplankton data were point features, there were large gaps along the study area that limited the accuracy of my interpolation. And also in reality, my data was collected along transects where ships pulled plankton nets below sea level to capture the samples instead of just sampling at a single point, and that just wasn't reflected in the data that I got. So this would have affected my bivariate relationships, and I think there may have been more negative relationships had my zooplankton data been of higher quality. And it's not that it wasn't of high quality, it was an extremely comprehensive data set. It's just it wasn't meant for the purpose that I used it for. And then the third major limitation is overall project scope. So there was 3 major issues here. The first is that the routes did not take into account other marine species that were similarly affected by ships. The second issue was that the computer I used for this project really limited the resolution of my analyses, which affected the applicability and accuracy of the results. 5 kilometers squared was just way too big of a grid cell size for this project. And then finally, there was a bias in the shipping data towards cargo shipping. So my results really say little about other shipping vessel types. So in conclusion, zooplankton makes up a large proportion of the ocean marine wildlife, and they're really important species in various or all stages of their lives. My project gave preliminary work for designing optimal shipping routes to avoid zooplankton, and it identified key limitations in the methods that I used. So hopefully the lessons that I learned from this project can help guide further work on the topic for both zooplankton and other marine wildlife, because the potential for multi-species approaches using similar methods is huge, even with like the data we just have. The NOAA's ecosystem monitoring program, for example, collects this type of data for dozens of species that you could do similar things with. And this could allow for more sustainable and data-driven decisions for our oceans. So this video is just about the NOAA's monitoring programs and the data they collect. I think it's a pretty inspirational data collection video. I have some more links to some resources, my references, my metadata, and I'll get my model up tonight if you want to look at it. And yeah, I just end with this quote from one of my favorite books, because I feel like I had very limited data and I made many mistakes in my methods, but here we are. Thank you for watching.